What's up, everybody? Herman here again. So today we're going to be discussing quantitative genetics. My partner Elizabeth, she discussed what would be most of the information that we'll be talking about in this presentation. Um, so I'm going to jump ahead to where she stopped, and we'll go from there. So let's jump right into it. Sorry, we had a technical error there. So I wanted to touch up on estimating response to selection. So we're gonna go over this a little bit. Um, so it's a critical component of quantitative genetics and breeding programs. It involves predicting the change in a population's mean value for a trait in response to selection. So we have the selection breeders equation, which is R equals H2 times S, where R is the response to selection, H2 is the narrow sense heritability, um, and S is the selection differential. And so we have the selection index theory, which involves the use of selection index to combine information from multiple traits and predict the response to selection. So we have genomic selection, which uses a genomic-wide marker data to predict the genetic merit of individuals and estimate the response to selection. Factors influence response to selection heritability. Um, the higher the heritability, the greater the response to selection. So that brings us to selection intensity. The stronger the selection, the greater the response to selection. Population size obviously plays a role. The larger the population size, the greater response to selection. There's genetic variation. The greater the genetic variation, the greater the response to selection. Um, Estimated responses to animal breeding. Um, it's critical in animal breeding programs to predict the genetic gain from selection. I know they do this a lot in plants as well, but in animals, they want to get certain traits. Like, I guess a great example would be like Wagyu meat. They try to get all the marbling in that steak, and I think like these cows, they have like security guards where they're protecting them in Japan. So they're high value. The genetic selection that's been bred, so they've been bred to have is set. Plant breeding programs, they predict the genetic gain from selection. So a lot of this is mostly for profit, but you get the idea. And we have conservation biology. Obviously, we want to protect certain species from going extinct, or generally they're difficult to raise. One of those was, or an interesting one to me, was condors. If you guys have never heard of that, that is a very cool thing that they did, conserving the condors in Arizona and California. Estimating response to selection can inform conservation efforts by predicting the impact of selection or on endangered populations. So, exactly what I said. Um, there are challenges and limitations, and I think this mostly affects like just genetic complexity. I know we've discussed just how complex genes can get, especially when chromosomes are aligning or separating. You have all these factors and variants that can happen or take place. The genetic architecture of traits can be complex, making it challenging to estimate response to selection. So it's just that I guess I wouldn't say, well, I guess we could say randomness and just the great variance that they have. Environmental effects can influence the expression of traits, making it challenging to estimate response to selection. An event could happen in a population and it could greatly affect, selection. The quality of data also used to estimate response to selection can impact the accuracy of the predictions. Let's jump into the next topic. So statistical tools. Quantitative genetics employs various statistical tools to analyze and understand the relationships between genes, environment, and phenotypes. We have the regression analysis, which is a simple linear regression. It models the relationship between a dependent variable and a single independent variable. And then we kind of make that a bit more complex with the multiple linear regression. It models the relationship between a dependent variable and multiple independent variables. Correlation analysis, Pearson's correlation coefficient. It measures the linear relationship between two continuous variables. Spearman's rank correlation coefficient. It measures the relationship between two ranked variables. ANOVA, analysis of variance. We have one-way, two-way ANOVA, and one-way it compares the means of two or more groups. Two-way compares the mean of two or more groups while controlling the effect of another variable. QTL mapping is a great tool. I've seen this before in it was molecular biology, I believe. It identifies, or mapping QTL, it's interval mapping. It identifies QTLs by analyzing the association between genetic markers and phenotypes. Composite interval mapping, it combines interval mapping with multiple regression to identify QTLs. GWAS, genome-wide association studies. We have case control studies. It compares the genetic variance of individuals with a specific disease or trait to those without. Quantitative trait, genome-wide studies. It analyzes the association between genetic variants and continuous traits. Heritability estimation, I believe Elizabeth touched on this a little bit. So the narrow sense heritability, it estimates the proportion of phenotypic variation due to additive genetic effects. The broad sense heritability, it estimates the proportion of phenotypic variation due to all genetic effects. Bayesian methods, it uses Bayesian statistics to estimate genetic parameters such as heritability and QTL effects. Bayesian QTL mapping uses statistics to identify QTL's Bayesian statistics. Sorry. So we're back to epigenetics. Epigenetics is the study of heritable changes in gene function that occur without a change in the underlying DNA sequence. So these changes can be influenced by various factors, and they include environmental conditions, lifestyle, experiences. So it's not necessarily a genetic or genetically based modifications, but we do have several mechanisms. One of those is DNA methylation, the addition of a methyl group to DNA. This typically results in a gene slicing. Histone modification, the addition of various chemical groups to histone proteins. This can either relax or compact chromatin structure. Non-coding RNA-mediated regulation, the use of non-coding RNAs such as microRNAs and siRNAs to regulate gene expression. Chromatin remodeling, that's the reorganization of chromatin structure. This either facilitates or inhibits gene expression. We have inheritance, epigenetic inheritance. So there's mitotic inheritance, meiotic inheritance. In mitotic inheritance, epigenetic marks are maintained during cell division, ensuring that the daughter cells inherit the same epigenetic information. In meiotic, obviously, it's marks that can be transmitted to offspring through the germline, influencing development and disease susceptibility. So environmental epigenetics, I've discussed this before several times. Exposure to toxins, it's one of my favorite. Pesticides, heavy metals, all this can alter epigenetic marks. Diet and nutrition, exercise. It's possibly the most prevalent, especially here in America. Dietary components such as folate and other methyl donors can influence epigenetic marks. Stress and behavior, these are lifestyle or experiences. Stress and behavioral factors such as maternal care can shape epigenetic marks. Epigenetics and disease. So cancer, I don't think I've talked about this before. Epigenetic alterations are a hallmark of cancer, contributing to tumorigenesis and tumor progression. Neurological disorders, epigenetic changes have been implicated in various neurological disorders, including Alzheimer's disease and Parkinson's disease. Metabolic disorders, epigenetic alterations have been linked to metabolic disorders such as obesity and type 2 diabetes. I would even dare say type 3, which is Alzheimer's. But yeah, that's epigenetics. Again, we've discussed it several times. I think the most important thing to take away here is the effect of environment. I guess even we could even include psychological factors here, but all this affects epigenetics. So thanks, guys. I believe that was the last topic I had to discuss. I hope you guys enjoyed this collaborative effort. We have a few more presentations to do, but it should all work out. See you guys in the next one.